Hello student, I am Dr. B.K.Barun from Medico Point, Patna. The today class is the biomolecules, that is the carbohydrate. Actually biomolecules are two types, that is macromolecules and micromolecules. The macromolecule means size is more than 1000 or 10,000 and macromolecules means size is around 18 to 800 Dalton. So the carbohydrate, protein, lipid, nucleic acid, these are the macromolecules. But lipid is exception. It is not as strictly macromolecules. But amino acids, simple sugar, nucleotides are the macromolecules. So first I telling the carbohydrates. So what is carbohydrate? So carbohydrate is the hydrates of carbon. That means it consists carbon, hydrogen and oxygen in the ratio 1 is to 2 is to 1. But this is old definition. It is hydrate of carbon. The new definition is that the carbohydrate is a polyhydroxyaldehyde or ketone derivatives. That means it has more OH group with aldose sugar. That is the aldehyde group. And if it is a ketonic group, so this is ketose sugar. So new definition of carbohydrate is it is a polyhydroxyaldehyde or ketone derivatives. And carbohydrate is divided into three parts. That is the monosaccharide, oligosaccharide and polysaccharide. Monosaccharide means it is simple sugar, that means not further divide because it is simplest sugar. And the monosaccharide start from 3 carbon to 7 carbon, that means triose to heptose. And there are different type of example triose to heptose in the account of aldose and ketose sugar. Such as in case of triose, the glyceraldehyde is aldose sugar, but dihydroxyacetone is ketose sugar. N is equal to 4, that is the tetrose. Erythrose is the aldose sugar, but erythrulose is the ketose sugar. And N is equal to 5 because general formula is CnH2nOn. So ribose is the aldose sugar, but ribulose is the ketose sugar. But deoxyribose is derivative sugar because this deoxyribose present in the DNA and ribose present in RNA. And in case of hexose, the glucose, galactose, mannose are the aldose sugar, but fructose is ketose sugar. And in case of heptose, heptulose is ketose sugar, but pseudoheptulose is heptulose is aldose sugar, but pseudoheptulose is ketose sugar. So these are the monosaccharides, and it is simplest, not further divide. And in oligosaccharide, disaccharide, tetra, penta, these are the oligosaccharides. Mainly in the disaccharides, sucrose, lactose and maltose are the main disaccharides. And other disaccharides such as isomaltose, cellobiose, trehalose is also disaccharide. So in case of sucrose, the bond is alpha 1 beta 2 diglycosidic bond, and it consists glucose and fructose. And in case of lactose, the bond is beta 1 4, that is the between the galactose and glucose. It consists galactose and glucose in case of lactose, and it is the milk sugar, but sucrose is the cane sugar. And in case of maltose, that is malt sugar, it consists glucose and glucose, and the bond is alpha 1 4 diglycosidic bond. And other disaccharide, that is isomaltose, the bond is alpha 1 6, cellobiose bond is beta 1 4, and trehalose bond is alpha 1 1. This trehalose is mainly present in the hemolymph of insect, and it provides energy for flight. And in case of trisaccharide, that is glucose, fructose, galactose is present, that is the astacose, verbicose. These are the raffinose. These are the other oligosaccharides. It may be trisaccharide, it may be tetrasaccharide. Say raffinose is a trisaccharide. and astracose is a tetrasaccharide, verbicose is a pentasaccharide. And in case of polysaccharide, the polysaccharide is actually more than 6, 7, 8, 10 monosaccharide. And these polysaccharide is divided into two groups, that is homopolysaccharide and heteropolysaccharide. So homopolysaccharides includes the collection of similar type of sugar, such as the homopolysaccharide includes the starch, glycogen, inulin, and cellulose. And cellulose is the most abundant homopolysaccharide in the whole inverse, because the plant consists cell wall and cell wall consists cellulose. So these are the cellulose. So homopolysaccharide mainly a starch. A starch gives the blue color with iodine because it forms the helix like a structure. But cellulose does not give the blue color because it is a straight chain. But glycogen is a more branched. That is the branch chain, glycogen. And in case of glycogen or cellulose or inulin or a starch, their left end is non-reducing or right end is reducing, because their right end consists the aldehydic free group, so it is reducing, and left end is non-reducing. So this is different between a starch and cellulose. A starch consists many glucose molecules, and glycogen, starch present in plant, but glycogen present mainly in animal and some fungus. And inulin present in the root of dahlia. It consists the fructose. And this inulin also used in the kidney clearance test or nephric filtration test, because it is not absorbed in their body. And this inulin is very important, and starch, glycogen, inulin is a storage polysaccharide, but cellulose, hemicellulose, these are the structural polysaccharide. And in heteropolysaccharide includes chitin, pectin, bacterial mucopolysaccharide, chondroitin sulfate, dermatin sulfate, sialic acid, heparin, hyaluronic acid. These are the heteropolysaccharides, such as chitin. Chitin present in their cell wall of the fungus, and it consists N-acetyl glucosamine. And pectin consists galactouronic acid. And bacterial mucopolysaccharide consists NAG and NAMA, that is N-acetyl glucosamine and N-acetyl muramic acid. So these are the heteropolysaccharide. Chondroitin sulfate and dermatin sulfate, these are mainly present in cartilage, bone, joints. And heparin is anticoagulant. It is also heteropolysaccharide. And hyaluronic acid present in also in the joint, and it is a heteropolysaccharide. So these all are the heteropolysaccharides. Thank you.